Okay, so we were talking about the breeder's equation, and the response to selection is the narrow sense heritability multiplied by the selection differential. The selection differential is a measure of how stringently you are selecting in a population, and the response to selection is, well, what you would see in the next generation. And this is actually data from spring of 2012 and summer 2013, where I had them do what I've asked you to do, which is bring in your height and the height of all of your full siblings. There are ways we could incorporate data from half brothers and half sisters, but that gets complicated, and I'm actually not entirely sure how. There is a way, but I've never had to know it. So yourself and any full blood siblings that you've got, and the mean height of your parents. And again, if you can't bring that data in for any reason, that's fine. I'm not going to knock points off your grade if you don't. And I'm not going to record anything other than a pair of numbers. There's no identifying information at all. But this is, and by the way, please don't make something up. I mean, for this to work, I need honest data. And I don't think most of you would, but there's always some joker who's going to claim that his parents are like, you know, two standout basketball centers or something like that. Allegedly, that's Yao Ming came from parents that had been selected by the Chinese government to marry each other because they were both really tall. No idea if that's true or not, but you hear rumors that Yao Ming is the product of eugenics. Anyway, so that was my data for the summer of 13 and the spring of 12. And I did what I just showed you and graphed mean parental height on the x-axis versus mean offspring height on the y. And you can see there's a pretty good positive correlation. And the slope of that line happens to be 0.915. Now, the accepted value is 0.7. When you do this in typical human populations, you get about 0.7. And I've added nearly 10 years of data since then, and I think it is closer to 0.7. I've got a much, much bigger data set than this. And of course, it's going to get even bigger yet once we've added your numbers to it. But I've got data on something like 150 people now. And I think the slope is closer to 0.7, but this is how we estimate narrow sense heritability. It's the slope of the linear correlation between mean parental and mean offspring phenotypes. Yes?
Can we also get data from our grandparents if we wanted, or since I'm an only child, I feel like?
Well, if you're the only child, then your height is the mean. Yeah, your height is the mean offspring.
But it's fine. Mean offspring height. My grandma was really tiny. I was like,
okay. Well, I don't know your family. I mean, that's actually true in mine. My dad's mother was about 5'2", and my dad's over 6 feet, and so am I. So sometimes that happens, but you're getting, my dad's mother would have actually survived things like the Great Depression. And I mean, you know, living on a farm in Iowa, you know, they grew a lot of their own food, and I'm sure some years there might not have been quite enough to go around. So you're getting to the stage where we have to reckon with different environments. I mean, most of my students come out of a kind of, you know, middle-class background. But if we start going back into earlier generations, we get people that were raised in physically and dietarily a different environment from our own. We get people that might have gone hungry part of the time. We get people that certainly would have, worked physically a lot harder than we generally do. Most of you folks don't plow fields with mules. So yeah, if we take it far back, we start getting environmental effects. We get that extra variable thrown into the mix. Okay. I mean, that would be interesting to look at if we could get data from like types of people back in like rural Arkansas in 1900 compared with it today. Sorry, I'm wondering if there's a way we can get that now. You wouldn't get it through the US Census because they don't record height, but I wonder if there's a way we could get very old hospital data or something like that. That would actually make for a really interesting lab, if I can find some data. Data. Yeah, that's why you have to hurry up in lab, because you've got to gather data. Get it? Okay, right. Here was some data. This is again from work on Galapagos finches. By Peter and Rosemary Grant. And they graphed in 1976, what those dark dots are showing is mean parent versus mean offspring beak size in 1976. That's the dark lines. And what you can see is that there's a strong positive correlation. The narrow sense heritability is about 0.8. After the drought, notice that the line shifted, but the slope is still about the same. The line shifted, but it didn't pivot. That suggests that, okay, we had a big environmental change, and what that does is shove the line over, but the slope is still nearly the same. So that suggests that we have a change in environment, but we haven't really had a change in genes here, that we're measuring something genetic. Okay, and I remember that in that drought, birds with small beaks tended to starve and birds with larger beaks tended to make it through. So when that line shifts over to the right, that dashed line is to the right of the solid line, that reflects the fact that the birds that survived the drought had larger beaks. But even though we had that environmental change and only the birds with larger beaks survived, we didn't change the fact that birds with larger beaks tended to have offspring with larger beaks, and birds with smaller beaks tended to have offspring with smaller beaks. Okay, we didn't change that relationship between genes and the trait. There's another complication with studying variance, and we're going to look at this in lab starting next week. Some of the variance in our traits might result from variation resulting from environment-gene interactions. You can have genes that are turned on or turned off or modified by some environmental cue. A fun example of this is Siamese cats. You've heard of Siamese cats, yes? Okay, what makes Siamese cats look the way they do is that they've got a gene that affects their production of skin pigment. And it causes them to produce skin pigment only in areas of the body that are cool. So think about it. The parts of a cat's body that are cool tend to be the same as the parts of your body that cool. You know, if you walk outside on a cold day, what are the parts of your body that feel cold the first? Right, ears, nose, probably face, hands, feet, right? Yes? It's the same in a Siamese cat. The cooler parts of a cat's body are its ears, its face, its feet, also its tail. And those are the parts of a Siamese cat that show color. Right? Yes? And I haven't really had the chance to observe it for myself, but my wife had a Siamese cat when she was little. And the cat spent a lot of time outdoors, and the cat did get noticeably darker in winter. You're nodding up there. Have you seen this too?
Yeah, I have a Siamese cat, and when I got her, it was like spring,
Mhm.
and well, she ran away. When it was like October, but she was like noticeably way darker.
Okay. She got way darker in winter. Okay. Yeah. Yeah, if the cat's whole body is cooler, then it's going to start producing more pigment and you're going to get a darker cat. You could take a Siamese cat and shave a patch of fur off the cat's back and tape an ice pack to the shaved patch. And you would end up with a seriously pissed off cat that's going to crap in your shoes. But in theory, you would end up with a Siamese cat that's where the hair growing in where you shaved it would be darker than the rest. You could even make kind of, tattoos on your Siamese cat if you just shave parts of the hair and chill the exposed skin. There's a breed of rabbit that does the same thing, by the way. If you've ever seen a Himalayan rabbit, they also have dark pigment only on the cold parts of the body. So that's a simple example of genes turning on because of an environmental cue. And if we were measuring the darkness of cat color in a cat population, some of that variance might be due to genes, some of it might be due to diet or something in the environment, and some of it might be due to genes getting affected by the environment. And we call that VG cross E. So total phenotypic variance is genetic variation plus environmental variation and variation of genetics cross environment. Yes?
So what's the difference between this and epistatic variance?
Okay, epistatic variance is one gene modifying another gene. So with Siamese cats, you do have epistasis because the Siamese allele is modifying where the pigment can go. You also have VG cross E because how strongly the Siamese allele modifies where the pigment goes depends on the environment that the cat is in. So in the Siamese example, we've actually got both. And you can really see this in a phenomenon that we call norms of reaction. And one of the things that you might not have taken away from genetics class is that very often a genotype doesn't specify exactly what the phenotype is going to be. Like when Mendel is crossing his pea plants and he's got purple flowered and white flowered peas, all of his peas that had the dominant purple allele had perfectly purple flowers. And in that case, the genotype tells you whether you're going to have purple flowers or white flowers. But very often what the genotype does is determine a range of phenotypes. But exactly which one the organism actually gets depends on the environment that you put it in. The fact that phenotypes vary with environment we call phenotypic plasticity. And given a certain genotype, we can actually draw a graph showing a relationship between phenotype and environment. And we would call that relation a reaction norm. So just the general fact that phenotypes can vary with environment is called phenotypic plasticity. On the other hand, if we measured a bunch of cats for average amount of dark pigment, and we graphed that against temperature that they were being raised in, the relationship between how dark a cat looks and how cold its environment is would be a reaction norm. A reaction norm is a description of what type of phenotypic plasticity you have. Give you a second to get that down, and I'll show you some examples. They're closer than perhaps you might think. All right, you with me there? Rotifers are virtually microscopic animals. They are in fact animals, but they are the size of some of the larger single-celled critters. You find them in enormous quantity in a lot of freshwater environments. There's also marine environments. And on the left, you've got a rotifer in the species Brachionis caliciflorus. The rotifer would normally have some cilia sticking out of the wide end and would be using those to swim. But yeah, it looks a little bit like a stemless wine glass. Here's the thing, Brachionis is not very big, and there are predators that would happily eat it. Notably a predatory rotifer that's a lot bigger called a splankna. And if you grow Brachionis and a splankna together, Brachionis will grow spines. If you grow Brachionis and expose it to water that a splankna has lived in, Brachionis will grow spines. Brachionis can respond to some sort of compound. I'm not sure we know what it is, but some sort of compound that a splankna gives off. And Brachionis will grow spines, or if it detects that particular compound, spines, making it much harder for a splankna to manipulate it and eat it. That's your phenotypic plasticity. All right, so there's not a gene for spines in Brachionis. There are genes that can be activated to grow spines or not activated to not grow spines based on signals from the environment that Brachionis is in. That's phenotypic plasticity. Daphnia. Did you guys work with this in your like 1400 or 1440 or 41? Anybody play with Daphnia?
I think it's
Okay, because we do Daphnia experiments in 1400, but I don't know what you guys do in 40 these days. Anyway, Daphnias, the common name is water flea. They look like little bouncing dots when seen with the naked eye. They're very common in freshwater and an important link in the food chain. And on the right is a fairly normal Daphnia under the scanning electron microscope. On the left is a Daphnia that's been grown in the presence of predators, and that Daphnia has sprouted a helmet. And it's also grown a much longer tail spine. And this is because these Daphnia were exposed to some sort of chemicals produced by predators, the common name of which is chiromones. And this is phenotypic plasticity as well. The phenotype of Daphnia is altered by the environment depending on whether the chiromones are present or not. In this specific case, you'll see this called an inducible defense. You can have it for traits that are not defensive in nature. But for the rotifer we looked at and for Daphnia, those are specifically defenses against prey. Presumably, it's energetically expensive to grow spines like that, and it might make it harder to swim. So it's not necessarily adaptive to have those spines on all the time. But the genes for spine production can be switched on if a predator is present. Isn't that cool? I was impressed. All right, here's another one that's not an inducible defense. This is a European plant called water crowfoot. It's Ranunculus aquatilis. It's a relative of buttercups. And it grows half submerged. The part of the plant that is above the surface of the water grows broad leaves. And the part of the plant that's below the surface of the water grows leaves that look like bundles of branching filaments. I'm guessing that leaves like that have lower drag, meaning that if the current gets fast, there's less of a chance that that plant's going to get uprooted and dragged downstream. I don't really know if that's been tested, but it's another great example of phenotypic plasticity. You can get different phenotypes, even on the same plant, based not only on genes, but based on signals from the environment that modify the expression of genes involved in laying down new leaves. This is actually something that Lamarck mentioned. And there are many of the examples that he listed, Lamarck did in his book, are perfectly good examples of phenotypic plasticity. It is quite possible for one organism to show multiple phenotypes, depending on what environment you put it in. This kind of thing happens all the time. Hey, I'll give you another one. I took this photo a couple of years ago at one of the Kroger stores in town. I can't remember if it was working class Kroger on Oak or a bougie Kroger over on Salem. I think this might have been bougie Kroger. This was in the spring, and they were selling hydrangeas. And this is a very common garden plant. You see them in gardens all over town. I noticed there were some nice hydrangeas in one of the houses as I was driving into work this morning. By the way, I'm sorry I'm late. I got right up to the railroad tracks, and the gates came down and the lights started blinking, and we sat there for quite a long time, and then the gates came back up. So evidently we had a ghost train passing through town. You ever had that happen to you? One of these trains that nobody can actually see, but the bells start ringing and the gates come down?
No, but we, one time here, like one of the first day of my freshman year of school, the gates almost came down on top of us.
Mhm.
That was nice.
Right. Yeah, well, sometimes it's because there's a train and sometimes there's not. Anyway, this is another great example of phenotypic plasticity, and it's one you can see around town or even in garden stores in the spring. Hydrangeas produce blue pigment if the soil is a little bit acidic, because acid soil has free aluminum ions, and the plant can take those up, and they're used somehow in the production of pigments. I assume as an enzyme cofactor. I don't really know how. But in slightly acidic soil, plants can absorb free aluminum. In alkaline soil, what happens is that aluminum ions tend to stay bound to organic compounds in soil. The aluminum tends to stick. Hydrangea can't take up aluminum, and so hydrangea can't make blue pigment. It can only make pink. So you can actually change the color of your hydrangeas by modifying the soil. I'm not quite sure how, but adding iron tends to acidify your soil. So you can turn pink hydrangeas into blue by burying some rusty nails by the roots. Or so I'm told. I haven't tried that myself, but maybe I'll sneak into this nice lady's backyard and do the experiment. So that's another case of phenotypic plasticity, and one that you can see around here quite readily. I will note that on the right, you see there are some white hydrangeas. That's not phenotypically plastic. Some strains of hydrangea carry alleles for no pigment. They're not carrying pigment producing alleles at all. So white hydrangeas are white because they don't have alleles for pigment production. Blue and pink do have alleles for pigment production, but soil conditions determine what pigment they're actually going to be able to produce. So that much is very definitely phenotypic plasticity. Okay, here's an example of a reaction norm. This is data from fruit flies. And up there at the top, there's a black line that's labeled wild type. Actually, this is a German graph, so it's Wildtyp, but it's wild type. And what they've done is calculated the Anzahl der Facetten, the number of eye facets. Remember that flies have compound eyes. They've got these big eyes that are made up of a bunch of little receptors stuck together. Yes, right? Okay. So they probably got some poor grad student to sit there and count the number of facets in the eyes of a whole bunch of fruit flies, because grad students have an infinite capacity for donkey work. And they got that black line that you see up there at the top. When you grow your fruit flies at 15 Celsius, they'll develop over 1,000 facets per eye. When you grow them under warm conditions, they might only get about 800. Okay, that's phenotypic plasticity in general, that the number of eye facets that a fruit fly has is a function of the temperature in which it lives. And the equation for that black line specifically would be our reaction norm. Okay, the reaction norm is the mathematical description of phenotypic plasticity. Given a certain genotype, what phenotype do you get compared to what environment do you get? All right, am I making sense there? There are fruit flies that have what are called bar eyes. Very small eyes that instead of being shaped like big globes are just shaped like little strips, little bars. And it turns out there are two different fruit fly mutant alleles called infrabar and ultrabar. And if you grow intrabar and ultrabar flies at roughly 17 degrees Celsius, both of those will give you flies with maybe 180 facets, maybe 200 or a little bit less. But the difference is that if you grow infrabar at higher temperatures, you get more facets. If you grow infrabar flies at 30 Celsius, you get about 300 facets. If you grow ultrabar flies at 30 Celsius, you get maybe 60. So what's the difference between infrabar and ultrabar? It's not necessarily the phenotype, because if you grow them both where those lines cross at about 17 Celsius, you get the same phenotype. The difference between them is they give you different reaction norms. The phenotype responds in different ways to environmental change. So with infrabar, if you grow these flies at warmer temperatures, you get more facets, and that's shown by the blue line. With ultrabar, if you grow them in higher temperatures, you get fewer facets, and that's shown by the red line. Okay, so those red lines are depictions of reaction norms. And reaction norms are the way we describe phenotypic plasticity. All right, is everybody so filled with knowledge that it's practically dripping from your ears? Yes.
I vaguely remember back in genetics talking about phenotypic plasticity, but also there's another type of epigenetic development in which the sort of was activated in infancy, but after maturity it could not be reversed. Was that something?
You can get, okay, the question was about epigenetics. Okay, you can get this thing happening where, okay, we used to think that Lamarck was totally wrong and there was no way the environment could modify your DNA that you passed on to the next generation. We now know that's not entirely true because you can stick methyl groups on your DNA. Methyl, right, just CH3, right, 1 carbon, 3 hydrogens. And methylation of certain DNA bases can affect gene expression. And those patterns of methylation can actually be inherited for at least a few generations, probably not indefinitely. But it is in fact possible to take a population, put it through some kind of environmental change. As a result, some of their genes get methylated, and then the methylation patterns will get passed on. Remember I mentioned the Dutch hunger winter, where people who were kids in the winter of 44, the winter of 44, spring of 45, grew up shorter than they would have? Well, it actually turns out that their children are shorter than average, even though they're growing up in modern the Netherlands and not undergoing starvation at all. So there actually does seem to be something that got acquired by the Dutch who survived the hunger winter and passed on to their kids after the fact. And that can look like Lamarckian inheritance, although how important it is in the grand scheme, I'm not sure. Those methylation patterns aren't permanent. But this can be a way in which populations can adapt, at least temporarily, on a very short time scale. Exactly what you were talking about with epigenetic effects in infancy, but not showing up later.
Oh, no, they would be, like the genes would be turned on in infancy, and then after that phenotype is present in maturity, those effects can be reversed.
Okay, they can?
They cannot.
They cannot. Okay, so you get genes turned on by the environment in infancy, but even after there comes a point where the genes are still going to be expressed. Okay.
So if you get like the same species of like wildflower that's growing like temperate forest or whatever.
Okay.
They are phenotypically different than the same species that's growing in like mountainsides.
Okay. All right. I'll let me look into that. I see what you're getting at now. You have, so if I take some wildflower seedlings and I put some in forest and some on a mountainside, the different environments will cause them to develop different traits. And that's reversible when the plant is young, but it stops being reversible once the plant's reached maturity. Okay, I'm not, Gesundheit, I'm not specifically familiar with that, but okay, clearly I need to go look that up. Sometimes I've got homework too. Okay, but there is a nifty example of this from way back in 1948. Some botanists in California went out and collected cuttings from wild yarrow plants. Yarrow in the genus Achillea, one particular species, Achillea millefolium, is edible and sometimes used medicinally as well. I used to grow it a long time ago. And so they went out and collected cuttings from 7 wild plants and grew them in the same garden at Mather, California. And this is an example of what we've called a common garden experiment. This is what they got. Now, all of these plants are growing in the same garden. They're growing in as close to identical environments as we can reasonably get. So any differences in height must be genetic because there's no environmental variance, right? And clearly there's a lot of genetic variance because some of these grew really short and some of these grew really tall. All right, with me there. When they grew cuttings from the same 7 wild plants at a different garden at Stanford, they got this. I should mention that the Mather Garden is pretty high up in the Sierra Nevada mountains. Stanford in Palo Alto, which I'm supposed to hate because I graduated from Berkeley. Berkeley Stanford is a, by West Coast standards, that's a major football rivalry. By SEC standards, it doesn't even freaking register. Because like the SEC is the only place where you find football rivalries that have been refined to a high degree of sheer hatred. But anyway, Stanford is down close to sea level, down in the San Francisco Bay. Mather, the first place, is up in the mountains. And when they grew cuttings from those 7 wild plants, they got this. So they're all growing in the same environment. So again, with no environmental variance here, the differences have to be genetic. With me there? But let's put those together. And remember that, right? Plant A at Mather and A at Stanford are genetically identical. They are both cuttings from the same wild plant, right? So every plant is genetically identical with the one immediately below it or above it. So, you know, if that plant is genetically identical to that plant's genetically identical to that one's identical to that one to that one, and so on. And remember that every plant at Mather and every plant at Stanford are grown in identical environmental conditions. What's making these plants different is their norms of reaction. Notice how plant A grew the tallest at both Mather and at Stanford. Plant B was second tallest at Stanford, but really small at Mather. All right, you see what I'm getting at? Plant E was second tallest at Mather, and not nearly as tall at Stanford. Let me see. You know, in fact, plants B, D, and F all grew significantly shorter at Mather, but relatively grew much taller at Stanford. So what's different about these 7 wild plants? Well, they are genetically different. They were grown in different environments, but what's really different about them is not so much that they're just genetically different, it's that they've got different norms of reaction. Okay, one more thing about heritability. Heritability, you can look up heritabilities for various traits of interest to animal breeders. But heritability can actually decrease in a population with time. Back in 1985, they took some chickens and tried selecting them for increased meat production. For the first 4 generations, meat production showed a narrow sense heritability of about 0.5. But as the experiment went on, narrow sense heritability started going down. Why would this be? Remember that narrow sense heritability is based on variance, right? Yes? Is based on the range of variation in a population. After a population's been highly selected, it doesn't have much variance at all. If all of your chickens are big-time meat breeders, then you may not have much variance in the trait. And if there's not much variance in the trait, then heritability goes down and may actually just end up being completely useless. And if narrow sense heritability goes down, what that means is that you're losing the ability to improve any further. You can select chickens for more meat if you want to, and it'll work for a while. Eventually you start running into limits. One of the limits being that if you don't have any variance in the population at all, you don't have anything for selection to work on. Another one of the limits being there are physical limits to just how big you can get a chicken before the poor thing just starts collapsing. The chickens that are bred for meat production today are actually a hybrid variety called Cornish cross. And they're slaughtered at a young age, but they're slaughtered about the time that they start dropping dead anyway, because they've been bred for so much meat that their circulatory system simply can't keep up with their body size. You know, by the time you're slaughtering your meat chickens, they're basically all cardiac patients. Because the heart just cannot keep up with the metabolic demands of all of the muscle that we bred them to have. Yeah, Cornish cross die pretty young. I mean, the chicken farmers that I know do try to give them a good life. I actually know several people that grow chickens organically, and they do try to treat them well, but they don't live. Yeah, they don't live very long. They have been bred for so much meat that they're starting to run into serious problems with survival. So there's limits to just how much change selection can cause, at least over the short term. And as you select your population more and more stringently, the total variance in the population gets less and less. Remember, if you're a chicken breeder, you do not want chickens that are just all over the freaking map. You want lots of chickens that will all produce lots of meat according to a predictable timetable, right? If you're a horse breeder, you do not want, you know, a population of horses that include Shetland ponies and Clydesdales and things like that. You want a whole bunch of horses that can all run really, really fast and win the Kentucky Derby, right? So if you are a professional at this, you probably don't want variance, because variance, you know, unpredictability is bad for business. There's a song from a musical called The Fantastics. It's something like, plant a radish, get a radish, never any doubt. That's why I like vegetables, you know what you're about. Plant a cabbage, get a cabbage, not a sauerkraut. That's why I like vegetables, you know what you're about. You know what you're gonna get. So if you're a breeder, your ability to select depends on there being variance in the population. But your ultimate goal is a population that doesn't have variance, where every bird is going to grow into exactly the same kind of chicken that produces exactly the same colossal amount of meat. And where every horse becomes a thoroughbred racehorse that can potentially win the Kentucky Derby. And where every cherry tomato produces large numbers of cherry tomatoes that are easy to pick by a mechanical harvester, and so on. Yes?
So in this case, the inbreeding coefficient would increase and heterozygosity would decrease.
Yeah, heterozygosity is going down because of selection. So the inbreeding coefficient is probably going up. But as heterozygosity goes down, that means that phenotypic variance is going down. And that means narrow sense heritability goes down. There was a study in 2002 of behavioral traits in German Shepherds. We know that we can breed dogs that have certain behaviors. One of my professors in grad school had a Shetland sheepdog. And as far as I know, that dog had never seen sheep in its life, but it still had very strong herding instincts. She would let it out to play, and the local kids would play on her front yard, and that dog would run around trying to herd the kids. It was rather adorable to see. And actually, if you own a Sheltie, you have to let them go out and herd something, because they'll go crazy if you don't. Shetland sheepdogs are very smart, but you have to give them something to do or they start going insane. Newfoundlands are naturally attracted to water. Pointers have been selected for strong instincts to point towards game. Shelties and other herding breeds like corgis have these very strong herding instincts. We know that we can select dogs to have certain behavioral traits, but within a certain dog breed, these behavioral traits have very low heritability because they have very low variation, right? You don't want to breed 2 champion German Shepherds and get a Yorkshire Terrier. You want to breed 2 champion German Shepherds and get more champion German Shepherds, right? Because that's how you make your money if you are a breeder of champion German Shepherds. So because of many years of selection, and because of limited genetic diversity in the German Shepherd gene pool, within that breed, heritability of behavioral traits is very low. We really can't do much more selection for behavioral traits in German Shepherds because they already don't vary very much in their behavioral traits. And that should be a caution to you, because remember that all this talk of narrow sense heritability makes no sense without reference to the underlying population that you measure it in. All right, if you're measuring a population of mutts, you'd find a lot of variability and probably a pretty high narrow sense heritability for traits in a population of mixed breeds. If you're measuring a population of only purebred German Shepherds, you'd find very low heritability because they don't vary very much. They're not supposed to. That's not what breeders want. Breeders want very much the same dog over and over again. So again, a trait can be inherited genetically and still have low heritability. So if German Shepherds aren't variable, then heritability of traits might be low. If you look at the total spectrum of dog breeds, or if you look at a bunch of mixed breeds, if you look at a bunch of, you know what Heinz dogs are? Heinz, they're a company that makes ketchup and steak sauce and things like that. And their slogan used to be 57 varieties. So a Heinz dog is a dog whose ancestry contains about 57 varieties. It's a completely mixed breed. So if you have a bunch of mixed breeds, if you have a bunch of Heinz 57 dogs, you might find a lot of variance and you might find high heritability for a lot of traits. If you're studying a highly inbred and not variable population of pedigree dogs, heritability might be very low. So heritability is meaningless except in reference to the specific population in which it's measured. Okay, does that make sense? Where this is getting back to my original attempt to debunk that book, The Bell Curve, is that you can measure IQ, and it does show a pretty high heritability. It's about 0.8. The authors of the bell curve use that to claim that your intelligence is basically determined by your genes. And if you are in a population that has these genes for lesser intelligence, which by pure coincidence, of course, is black, then you're going to be born with lesser intelligence and there's nothing you can do about it. The reason why that's a bowl of crap is that heritability is not the same thing as genetic determination. You could have a trait that in some populations has high heritability and in other populations it doesn't. Or you could have a trait that has high heritability and yet is strongly influenced by the environment. You know, intelligence is probably even more affected by the environment that you're in than rotifer spines or water crowfoot leaves. You cannot take heritability as some sort of inflexible determination of how much of a trait is governed by genes, because that simply isn't what heritability is. It's useful, but you can't use it in the ways that the bell curve does. Nor can you usually connect a specific gene to a quantitative trait. And this was a book that came out in 2004 called The God Gene, arguing that religion was ultimately hardwired into your genes. The book is also crap. And the book was basically one finding spread out over 250 pages that it really didn't need. One of the findings of that identical twin study that I mentioned is there is no heritability for the specific religion that you are. There's no genes that will make you more likely to be a Baptist than a Buddhist. That's entirely due to culture. If you're raised a Baptist, you'll probably be a Baptist, at least until recently. I mean, now with the internet, you can find out about so many new religions that your grandparents never heard of. So you could be raised a Methodist and end up an Asatru, or raised a Baptist and end up a Wiccan or something like that. What's that? Or a Scientologist. But up until fairly recently, people's religion was pretty much determined by how their parents raised them. Yeah, my grandfather was a Baptist going back many generations, and he only became a Methodist to please my grandmother. He used to say that the saddest sound in the world is a Methodist congregation making a joyful noise unto the Lord. Methodists tend to be very sedate. Baptists get a little more enthusiastic. Anyway, the specific religion that you are has nothing to do with your genes, but there is a personality trait that psychologists can measure called self-transcendence. And self-transcendence does not depend on any specific religion, but you measure it by giving people a questionnaire where they tell you things like how often they feel a sense of wonder at the world. How often do they feel that ordinary life is not all there is? How often do they read religious books? Whether that book is the Bible, the Quran, the Bhagavad Gita, whatever it is. How often do they go to religious practitioners for guidance? Whether that's a minister, a rabbi, a kofi, whatever. How often do they pray? Whether it's to God, Jesus, Allah, Buddha, Odin, Freya, Shakti, Hathor, Rhiannon, whatever it might be. And so you can calculate people's scores on this personality test and call that self-transcendence. And that does have a heritability of about 0.4 in the populations where it's been studied. So there's no gene for a specific religion, but there may be some genetic influence on whether you go to church every Sunday and get really enthusiastic about praying and singing hymns, or whether you only go on Christmas and Easter and just because you're expected to. You know, there may be some genetic influence on whether you are a cement head or a God bother. All right, with me there. Hamer published this book arguing that there are alleles for a protein that's involved in neurotransmitter release in the brain. The gene is called VMAT2, vesicular monoamine transporter. And he argued that alleles of this gene were correlated with a high degree of religious or spiritual behavior. So if you have some alleles, you are more likely to be the church lady who's always praising Jesus for everything. And if you have other alleles, you go on Christmas and Easter and you kind of sit there and then you're done for the year. Sounds really cool. Unfortunately, it turns out that VMAT2 accounts for roughly 1% of the variance in self-transcendence. Let me show you real quick what that looks like. Let's go over here to the whiteboard and erase all of this. Erase it all. All right, let's just say we'll draw another graph. And we're going to measure on the x-axis, that is your degree of self-transcendence graphed against the number of people. So we have, let's say, self-transcendence varies, and let's say it varies along a bell curve. So we have a few people who just really don't want to be bothered. And we have a bunch of people who are kind of in the mushy middle. And we have a few people way over there at the right end of the spectrum who just won't shut up about it. Okay, we have the Jehovah's Witnesses doorbell ringers are over there on the right. Okay, with me there. It turns out that if you look at different genotypes of VMAT2, then one genotype will have a trait distribution like this. And the other genotype of VMAT2 will have a trait distribution that looks kind of like this. So if you have one genotype of VMAT2, you may be slightly more likely to be a God botherer. I'm sorry, I can't think of a better term, and I hope that's not offensive. What's that?
Book thumper, Bible thumper.
Okay. Well, again, this is independent of religion, so this could be a holy book thumper. You know, a Bible thumper or a Tripitaka thumper or a Rig Veda thumper or a Havamal thumper or whatever. If you have one of these genotypes, you might be slightly more spiritual than if you have the other genotype, but the difference is extremely small. The difference is only about 1% of the total variance. So even though Hamer was claiming that VMAT2 is the God gene, and people who have one genotype of it are more spiritual than people that don't, the truth is that the effect is absolutely tiny. The truth is that the effect is so small that we have no business calling this any sort of God gene. What's that?
I wanted to fit in the like standard error anyways. Like 1% difference in whatever.
Yeah, that 1% is so small, it's probably, it could well just be experimental error. I don't have the stats at my fingertips. But yeah, you're quite right. That's such a small difference that it could just be experimental error. It is, I mean, it's not impossible that genes could affect your perception of spirituality because, I mean, VMAT2 is a gene for neurotransmitter processing, and genes that affect the way your neurotransmitters work, they certainly can affect the way you perceive things. So it's not impossible that your genes could have something to do with your spiritual yearnings. But we have yet to identify any individual genes that have anything more than the most minor effect on human spirituality. If there are genes that affect human spirituality, there's a lot of them, and they probably do a whole lot more than just that. So getting back to the bell curve, I tell you what, actually, I have 11:36. It's been about an hour since our last break. Let's go ahead and take another break and what the heck, come back at 11:45. That's a 9-minute break. And in the remaining time that we have, I'm going to take the bell curve apart, or at least I hope I will. So we will stop the recording now.